Hi guys and good day everyone. Today I, Stay Sharp with metric number 205653 and my teammate Nisa Hadira with metric number 197562 are going to present the article titled Machine Learning Model for Lymph Node Metastasis Prediction in Breast Cancer using Random Forest Algorithm and Mitochondrial Metabolism Hub Genes. Our presentations will be covering 5 parts, which are Introduction, Materials and Methods, Results, Discussion, and Conclusion. Let's begin with a brief introduction about this article. To make it easier to understand the article, let us break the title into few pieces and look into every single one of them. There are 4 components. Firstly, is a tool involved, that is, machine learning model. It is a predictive model that learns using algorithms-based data. Secondly, the aim or purpose of developing this model is to predict breast cancer by analyzing the expression status of genes related. Thirdly, the technique involved in this tool, or in other words, the algorithm used in this model, is random forest algorithm. And lastly, the data we are going to analyze, which provides the information, is hub genes. Overall, the objective of this study is to develop a machine learning model based on RNA sequencing data using the random forest algorithm and hub genes. The aim is to estimate the accuracy of the breast cancer metastasis prediction model. This study will compare the accuracy of prediction models using total genes, functional group genes, and also hub genes. This study also identifies a suitable machine learning algorithm to predict the breast cancer metastasis. Okay, now let's go to the second part, materials and methods. There will be a total of 6 parts. Let's move to the first part, data availability. So, where do researchers get their data and what types of data they use in this study? The researchers first obtain single-cell RNA sequence data from the NCBI JAW database. From that database, they pick up one breast cancer patient and obtain 94 single-cell samples from the patient. And these 94 single-cell samples, they are composed of 2 groups, which is 56 metastatic samples and 38 non-metastatic samples. Both of the data is sequenced using Illumina sequencing. Next, how does the researcher process the data? First, the sequencing data of patients will align to a reference genome history 8 using an alignment tool called STAR. Then, the gene expression levels were quantified using history sequence and the expression data was normalized using TPM. And those genes with TPM value more than 1, they will be undergo further analysis. Next, the DE Sequence 2 package in R program is used to detect the differences in gene expression between metastatic and non-metastatic breast cancer. Those genes that exceeding the cutoff will be selected. Lastly, the functions of genes in metastasis were analyzed based on the information from Human Cancer Metastasis Database, or short as HCMDB. Let's proceed to the next part. With all genes selected just now, the researcher will analyze them with PCA and volcano plot. PCA is used to analyze the overall variation in gene expression patterns between the samples. And we can look at the figure at the right side. The result is visualized using PRCOM function and also GGBI plot package in R program. And on the right side, the volcano plot, it is to visualize the differently expressed genes. After that, the researcher clusters the genes and identifies hub genes using a method called Weighted Gene Co-Expression Network Analysis, WGCNA. It is a method that calculates the correlation between gene expression profile and construct a network based on the correlation value. Genes with higher absolute correlations, they will be clustered into modules, and from the modules, they identify the functional ones by performing hierarchical clustering. In the WGCNA method, the researchers will reduce the noise and errors with the help of a program called TOM, Topological Overlap Matrix. They will also use term-based dissimilarity measure and dynamic tree-card algorithm to classify genes with similar expressions into profiles. The gene significance is determined through the linear regression between gene expression and clinical trait, while the MS, module significance, they are incorporated clinical information by calculating the average GS of all genes in a module. Overall, to make it simple, WGCNA allows us for identifications of hub genes and also the integration of clinical information into core expression network. We have mentioned hub genes all the times, but what is it actually? They are genes that are highly associated with clinical traits, and they can be identified based on their connectivity and correlation with clinical traits. And how can we get hub genes? From the raw data, we align them, and those that are functional are grouped together and clustered as hub genes. What kind of tool can we use to do it? We can use the conceptual hub analysis tool, CHAT, and the Cytoscape software to visualize hub gene clustering. And to identify the function of the gene set, we can use David Webb tool. And lastly, the researchers will select a specific gene set that is related to mitochondrial metabolism and breast cancer metastasis for the prediction model. We have mentioned that the metastasis prediction model requires machine learning techniques to run them. So in this research, different kinds of techniques were evaluated. They were evaluated based on their accuracy, precision, and recall score. And the one that performed the best is random forest algorithm. And in these studies, they also include external dataset from 96 breast cancer samples that is obtained from the Cancer Genome Atlas for better validation. That is why the hub gene sets was utilized in the validation process using the RF algorithm.
Next, we'll move on to results, which is divided into 4 parts. 3.1 Difference between normal and metastatic breast cancer. 3.2 Weighted co-expression network construction and key model identification. 3.3 Gene ontology and pathway enrichment analysis. And 3.4 Estimation of prediction model. For the results of the difference between normal and metastatic breast cancer, 94 single-cell samples were isolated from a breast cancer patient. By using the GEO database and RNA sequence analysis, 56 samples were found to be metastatic, isolated from lymph nodes, and 38 samples were found to be non-metastatic, isolated from normal breast cancer. This figure shows the PCA plot, which is a plot that groups samples upon similarities. The figure shows the distribution of the metastatic and non-metastatic samples. Next, the analysis and selection of genes were performed. It was found that 2,683 genes exhibited crucial differences between metastatic and non-metastatic samples, as shown in the volcano plot of differentially expressed genes. From the 2,683 genes found, the top 20 regulated up-down genes were listed by comparing non-metastatic and metastatic breast cancer as shown in the table. As for the WGCNA and key model identification, there were 4 key steps done in order to determine the best module to be used. Firstly, a co-expression network was developed. This is visualized by the dendrogram clustering the 94 samples. Secondly, the self-thresholding power in WGCNA was determined. The scale-free fit index and analysis of mean connectivity of the self-thresholding power is shown in the graph. Then, the genes expressions were clustered. This is based on the dissimilarity measure of 1 TOM. Lastly, 8 modules were developed. This is by grouping the genes with similar gene expression patterns into 8 different groups or modules represented by different colors. This was calculated by the WGCNA function. It was found that the brown module was the best module to be selected, as it has the highest gene significance in the metastasis statement. Under gene ontology and pathway enrichment analysis, the brown module having significant activity of metastasis was utilized by performing functional genes assay. These genes were then grouped into 3 functional groups: biological processes (BP), cellular component (CC), and molecular function (MF). Then, these groups had undergone the Kyoto Encyclopedia of Genes and Genomes (KEGG) pathway analysis that identified that these genes were involved in ribosome, spliceosome, proteasome, and Parkinson's disease. These results also show that the genes in this module are involved in cell proliferation and cell migration. Lastly, the estimation of the prediction model was done. The total genes expression test with 16,482 genes, random forest algorithm used, 54 functional gene modules, and 3 key hub genes. The prediction model had been established. From all the algorithms, random forest algorithm had the highest test accuracy of 0.945 or 94.5% accuracy, with the train and test accuracy via external validation of 0.910 or 91%. The predictive model had been constructed. Now let's move on to the discussion. There are several setbacks discussed that were overcome in the study. Firstly, there was trouble in applying nuclear imaging data in a clinical setting. By building this prediction model, higher accuracy than nuclear imaging data had been estimated to predict the accuracy of breast cancer metastasis. Next, there was limited number of genes to be analyzed to profile cancer cells. In this study, the concept of functional group gene and hub gene was used that gives off higher prediction accuracy. In this study, the weighted correlation network analysis (WGCNA) was performed. It is a bioinformatics analysis method to identify functional genes and hub genes. It is able to search for a function related to a highly expressed gene. Hence, this represents a more detailed and reliable indicator for the prediction of breast cancer metastasis.
In conclusion, we have discussed the development of a highly accurate predictive model for breast cancer metastasis using the RNA sequence data and different machine learning algorithms. We also mentioned that the machine learning that is focused on nuclear imaging is still remain challenging. However, if we use genetic data from RNA sequencings, they will offer more precise patient information than image analysis. It is also proof that this model has a potential that is significantly enhanced prognosis assessment for breast cancer patients. By combining RNA sequence data and machine learning, we are able to achieve high accuracy in predictive modeling. Furthermore, if we combine nuclear imaging and RNA sequence together, they will even give us a better promise for practical use in clinical settings. That is all from us. Thank you.